All right, so good morning or afternoon, everyone. I guess it depends where you're joining us from across Canada right now. But welcome to the Canadian Grapevine Certification Network's webinar, where we're going to be focusing on optimizing the quality of Canadian wines in today's session. So today we're going to be learning about the final results of activities 12, 13, and 14 of the previous Grape and Wine Research Cluster. And you can see the research themes on the screen that I have here. And please note that the webinar will be recorded and it will be posted to our website and our YouTube channel afterwards if you wish to view it again or to share it with your colleagues. So I'll start today's session. My name is Darian Temporal. I'm the Executive Director of the Canadian Grapevine Certification Network, and I'm going to serve as your moderator for today's session. We are joined by three fantastic guest speakers. We have Dr. Debbie Inglis from Brock University's Cool Climate Enology and Viticulture Institute in St. Catharines, Ontario, Dr. Lihua Fan from the Agriculture and Agri-Food Canada Kentville Research Centre from Kentville, Nova Scotia, and Dr. Gavin Kernaghan from Mount Saint Vincent University in Halifax, Nova Scotia. So before we get into our feature presentations today, I'm going to provide a very brief overview of the Grape and Wine Research Cluster and the research themes it's focused on. And then we will have our presentations from our three guest speakers who are going to provide a detailed look at the research processes and results of their concluded projects. So this would have been from 2018 to 2023. And then at the end, we're going to hold a question and answer session. And this is where attendees, everybody here, can ask the speakers any questions they would like about the research at hand. So please feel free to type your questions in the Q&A section at the bottom of your screen, or save them, and then you can actually have the opportunity to ask them in real time to the speakers during the Q&A session. Just let us know if you want to do that. All right, so I'll move into my presentation then here. So first, the Canadian Grapevine Certification Network represents wine grape growing interests across Canada with a focus on managing and mitigating grapevine virus infections. So we're a national not-for-profit organization comprised of membership of 4 main grape growing associations across Canada, being the British Columbia Wine Grape Council, Grape Growers of Ontario, Council des Vignes du Québec, and the Grape Growers Association of Nova Scotia. And each member association elects 2 directors to sit on the board. And today our board includes all of the names that you can see listed on the screen here. The board has maintained a mandate that is twofold. So first, it's to advance the Canadian grape and wine industry by ensuring grape growers have a sustainable domestic supply of certified clean grapevine propagative material, including both rootstock and scion wood. And then secondly, we support and promote grape and wine research efforts across the country. So since 2018, when this first cluster was announced, we have administered the National Grape and Wine Research Cluster through Agriculture and Agri-Food Canada's Agri-Science program, which I'm briefly going to discuss in this next slide. So for those who may be unfamiliar, the Grape and Wine Research Cluster is a national research funding program that was funded in part by the Government of Canada, and from 2018 to 2023, it was called the Canadian Agricultural Partnership Framework. The cluster was made up of several grape and wine research-related activities conducted at government research centers, universities, and other industry locations across Canada. So the CAP cluster framework ended in March 2023. So after giving the involved researchers a couple of years, to tie up loose ends, potentially publish articles on their work, we're excited to finally be able to present the findings from the cluster in this mini webinar series that we've been conducting from spring of this year into the summer. So we're finally sharing the final research results from many of our CAP cluster research activities. And you can view many of them already on our website from what we've conducted webinars for in the spring. So as part of our efforts to promote final research results from all of the activities in the CAP cluster, we hosted the National Research Users Meeting back in April alongside the British Columbia Wine Grape Council and Agriculture and Agri-Food Canada. So attendees of that event spent the day at the Summerland Research Station or joined virtually via Microsoft Teams. That's what I did, being based in Ontario. And then throughout the day, attendees got to learn about the final research results from all of the CAP cluster activities that were conducted at exclusively AAFC research centers across Canada. So all the presentations were recorded, and those have been uploaded to our website and our YouTube channel. So the CAP cluster activities that we've been talking about are organized by theme as follows. So the research activities are broken down into several key themes that are listed here. The activities we're going to learn about in today's webinar is bolded here on the screen. Activities 12 through 14 fell under optimizing the quality of Canadian wine themes. So this theme explores several topics such as the development of Indigenous yeast for domestic wine production, field to winery management strategies for improving fermentation, efficiency, and tannin profiles in red wine, improved processes for sparkling wine production, and consistency in the marketplace. So before we get into the feature presentations, I also would like to let you know that CGCN is currently administering the Grape Wine Cluster under the Sustainable Canadian Agricultural Partnership, or SCAP framework is what we call it. So it includes 15 research activities being conducted once again at government research centers, universities, or private research facilities across the country. And it's organized into one of three themes or priority areas: climate change and the environment, economic growth and development, or its sector resilience and societal challenges. And we're currently in the middle of year 3 out of the 5-year cluster, and research summaries from years 1 and 2 are now available on our website if you'd like to learn about each of the activities in a little bit more detail in simplified language. So you can see that there. If you want to find this on our website, you can go to the www.cgcn-RCCV.ca, and you can hover over the research tab. It'll show you another menu, cluster updates and events. Click that, and it'll bring you to the page. So thank you very much for allowing me to share a little bit about CGCN and the Grape Wine Cluster. Ethan and I are both happy to answer any questions you may have. So without further ado, I will stop sharing my screen here. And I will introduce our first speaker of the day. So we have Dr. Debbie Inglis attained her PhD from McMaster University in biochemistry in 1995. After a 3-year stint in a biotech industry, she joined Brock in 1999 as an assistant professor in the Department of Biological Sciences, teaching in the Enology and Viticulture program. She took on the directorship of the Cool Climate Enology and Viticulture Institute, or what we call COVI, in 2008. She now holds the title of full professor and continues to be the director of COVI. Debbie's research program first focused on the issues of ice wine fermentation, but since has expanded over years to other red and white wine styles, along with biochemical issues in grapevine cold hardiness and more recently diagnostics for grapevine diseases. She's been involved in the grape and wine industry her entire life, as her family were grape growers in the Niagara Peninsula in Ontario, and her brother owned a winery. She herself is a second-generation grape grower, and she knows firsthand the issues facing the industry and is very happy to help solve them through research. So Debbie, please, if you could present your research on tannin alert.
Thank you for that introduction. Oops, sorry. Ooh, I've got a little bit of a glitch here. Hold on. It's not pulling up my presentation properly.
Of course, it always happens in the actual sessions.
Great. Even though we did just check this out.
We just practiced.
Yeah. There we go.
There we go.
Great. Okay, thank you very much. Everybody can see that okay?
It brought us into the industry challenge section again, but maybe if you click back a couple, perfect. We're good.
Got it. Okay, thank you so much. So I'm going to present today on activity 12 of the CAP, which was our tannin alert project, looking at a new winemaking tool to help manage skin and seed tannin in red wines. So what was the industry challenge and why did we embark on this project? Producing high quality red wines consistently year after year in a changing climate can be a challenge when you're in a cool climate area, such as the various regions across Canada. In making red wine, you need skin contact for color extraction. And in trying to extract color and tannin from the skin, we have to be mindful of not over extracting bitter tannins from seeds. This issue of not over extracting bitter tannins from seeds or having a lot of seed tannin can be amplified in cool climate regions such as ours. Also, in regions such as in Ontario, and I would say also in Quebec and Nova Scotia, probably less so in some of the warmer regions of the Okanagan, fruit does not always reach optimal ripeness each year. For red grapes, tannin ripeness may not be achieved, leading to wines of lower quality. Skin color may not be fully developed, and skin tannin may be too low if we don't get those really warm seasons to allow for full color and tannin from the skin to develop. Also, the extractable seed tannin may be too high, so the grapes may not form a hard seed coat over the seeds, and this makes the seeds softer, and we can extract just far too much tannin from the seed through the skin and seed contact during red wine production, and that can lead to bitter and astringent wines. So although we have very easily implemented ways to look at ripeness of the grapes from the sugar level regarding Brix measurements, or the acidity level regarding titratable acidity or pH, we're really lacking when we started this project for an easy way to assess the tannin level in the grapes. And of course, consumers demand a high quality, consistent product each and every year, despite the annual weather and vintage variations. In Ontario, there was a quote a couple of years ago from the LCBO indicating that they were looking for ripe, rich, and rounded red wines, i.e. not bitter and astringent red wines. And in some years, this requires greater intervention by the winemaker to ensure that they're producing that consistent, ripe, rich, and rounded red wine each and every year. And this can be a disadvantage in tannin development in cool climates. So what are tannins? What am I talking about when I'm referring to tannins? Well, tannins are made up of these phenolic compounds. Don't worry, this isn't going to be a chemistry lecture on phenolic compounds, but this is a component of tannins. They have this aromatic ring with this hydroxyl group, and all tannins are going to have a makeup of various phenolic compounds. These compounds are found in various food products, including tea, dark chocolate, grapes, and of course, red wine. They can greatly impact the perceived quality of food products. And in wine, tannins contribute to the color of wine. That's the anthocyanin pigment made up of phenolics. But they can also help stabilize the color of a wine. When tannins bind to those anthocyanin pigments, they trap them in a colored complex and help stabilize that. They can add structure on the palate to the wine, but when they're in too high a concentration, they can add to astringency and bitterness in the wine. And specifically, bitterness is usually associated with high seed tannin that gets extracted into the wine. So again, as I stated earlier, in the winemaker's toolkit, great ways to monitor and follow Brix development in the fruit, acidity through pH and TA, but we didn't have an easy way to follow tannin development to determine when grapes are at their best capacity for that tannin development and to harvest those grapes. So the winemakers are lacking predictive analytical tools that would allow them to better manage skin and seed tannin. And this was actually brought to my attention. It was over 10 years ago. And I'll just make a shout out to the late, great Paul Boss Sr., who in a discussion I was having with him, really encouraged me to figure out a way that we could bring tannin measurements to the industry. And then through discussions when Belinda Kemp joined us in Covey, we realized that really what the industry needed was a separate measurement of skin tannin versus what was happening in the seed tannin and how that was changing leading up to harvest. And then to let winemakers know when skin and seed tannin are at their optimal levels, or if they're not at optimal levels, is it the skin tannin or is it the seed tannin that needs to be addressed? So what's desirable is to have high skin tannin and low seed tannin. That allows for lots of extraction of color and to stabilize that color with the skin tannin and not to extract the bitter and astringent seed tannin. What's not desirable is when we have low skin tannin and high seed tannin. When you have low skin tannin, it means the winemakers are going to have to work extra hard to extract that tannin from the skin. And there's a worry that they may over extract the seed tannin and have to look at other ways to help manage that. So we started in our pre-harvest monitoring program in Covey to include monitoring for the tannin levels in grapes from veraison right through to when the grapes were harvested. So every week we went out to red variety plots and took samples of the red grapes. We separated out berries from the clusters, and then from the berries separated out seeds and skins. Then we extracted the tannin from the seeds and skins and measured that tannin value using an assay called a methyl cellulose precipitation assay. In the Tannin Alert program that developed from our ability to measure separately skin and seed tannin, there's a few steps, and I just want to walk you through what the program looks like. So we're following tannin ripeness of red grapes for 6 different red varieties from veraison through to harvest. We're measuring skin and seed tannin separately for each of those varieties. We would then take those values of the skin and seed tannin at harvest and develop a database that accounted for all of that data from every variety over multiple vintages and multiple locations for these grapes being grown across the region. Once we had all of that data compiled and it was a large enough data set, we could look at the distribution of tannin in the skin and in the seed, and we could categorize ranges for low skin tannin, medium skin tannin, and high skin tannin, and then similarly low, medium, and high seed tannin. And the idea behind all this is to optimize red wine quality by accounting for the tannin levels in grapes at harvest, and then using winemaking protocols to help optimize that tannin extraction, given what the ranges are in each variety. So we actually started this project a few years before we actually started the CAP program. It was funded through an NSERC and OCE grant that I'll note at the end. So our database now contains data from 2015 all the way to 2022, with 2022 being the last vintage from the CAP that we had funding to go out and do this large sampling. So we were sampling Cabernet Sauvignon, Cabernet Franc, and Pinot Noir. And each year we were sampling from 4 sites at a minimum per year. And in some years, we actually had 6 sites. For Merlot, Gamay, and Syrah, we sampled from 2017 to 2022, and we were sampling 1 site per year. There were variety site and vintage differences in the tannin levels in both skin and seed within a variety. And I did present in detail that information in the CGCN talk in 2022. So I decided this time I wouldn't delve into all of that variability, but really get into how we use the database and how the Tannin Alert program works. So in the Tannin Alert service, Once we had that database, winemakers can then bring in a sample to the Covey lab to measure the tannin in their grapes at harvest, to document whether and get a readout of whether the tannin is in the low, medium, or high range for the skin and seed. And then they could develop their winemaking protocols around that. So in a perfect year, what we would expect to see for skin tannin is through veraison, right through the harvest, we would see an increase in each week in the tannin development in the skin. And then subsequently in the seed, as the seed coats develop and get harder, we're not able to extract as much tannin from the seed coat. And so from the interior of the seed because of that seed coat. So we would expect that the tannin value in the seed, the extractable tannin, would decrease as we get closer to harvest. That was in a perfect scenario. What we found through all the data that we collected year in and year out over from 2015 through to 2022, we did not always see this trend, which really drove home the importance of having this tannin database and being able to benchmark the tannin values in your grapes at harvest against this database. So how would a winemaker use the tannin system to benchmark their tannin values against what is in the tannin database? So once we had all the tannin data accumulated, in a perfect distribution, it would look like what I have illustrated here, where we would have a distribution of tannin in either skin or seed, and we would have a peak value with most samples coming in at that value, but then we would have a tail off from that value that would give us a lower distribution range for the tannin, a higher distribution range of the tannin at the high end, and then the medium distribution range for the majority of the samples that we were collecting. I want to share with you what an actual histogram of this distribution looked like. And this was the histogram for Pinot Noir for both the skin tannin and the seed tannin. And they're looking almost like the schematic, almost like the perfect distribution here for skin tannin. And for seed tannin, we don't get quite the drop off at the tail end for the low level. And again, this is, I think, to be expected in a cool climate region, because often we don't get that complete seed coat hardening in the varieties at harvest time in our cool climate region. So from these types of distributions, we were able to mathematically calculate the low range of tannin, which is to the 33rd percentile, the medium range of tannin, which is from the 33rd to the 66th percentile, and then the high range of tannin, which is above the 66th percentile. And so I won't go through this table in grave detail, but you'll see for every red variety that we tested, for skin tannin, which is in the gray columns, we've got the range for low, medium, and high tannin that we measure in micrograms of a certain type of phenolic called epicatechin per berry. It's kind of our reference phenolic that we use. And then in the seed tannin, we also have the low, medium, and high range of tannin from the seeds for every variety. But when I started looking at this data, something really struck out to me for the Gamay. And you'll see for Gamay, even in the high range of the amount of tannin that's present in the Gamay, it's still relatively low compared to all of the other high-end tannin for the other varieties. And then similarly for the seed tannin, Gamay has much higher values in each category compared to the other varieties. And it got me thinking that, you know, if we only benchmark a Gamay sample against the Gamay database, it might give a winemaker a false sense of security that they've got a high skin tannin and low seed tannin, which would be true when benchmarked against other Gamay, but when benchmarked against other varieties, the high skin tannin would actually come in fairly low and against the full database. So because of this conundrum, I decided that it might also be worthwhile to compile a full database with all 6 varieties. And so I did that. We just pulled all of the data from all 6 varieties, from all sampling vineyards at harvest for all the years, and came up with this combined database for skin tannin and seed tannin. And then we could have a combined database to define what the low, medium, and high range is for both skin and seed. So just to walk you through quickly some actual data, if I took Gamay, the Gamay database, and I plot out here in all these beautiful colors, the low region for skin and seed tannin in green, the medium region in yellow, and the high region in red. And then I took a Gamay sample and we actually measured the skin and seed value for the Gamay. You can see that for that sample, it's reading 328 micrograms of epicatechin per berry for the skin, which would put it into the high category against the Gamay database. And similarly, the seed tannin would be in the low category. But when I compare those same values to the all variety database, you see that the skin tannin is actually coming in fairly low against the full database, and the seed tannin is coming in high. So why this is important, because if you were carrying out processes thinking that you had low seed tannin and ended up extracting all kinds of seed tannin through the winemaking process, in reality, that seed tannin compared to other varieties was actually at a fairly high level. And you need to be a little bit more cognizant of that high seed tannin and then work a little bit harder to extract the tannin from the skin to help stabilize color over time. Similarly, I just wanted to show you some data from another variety, Merlot. We had a sample come in, we benchmarked it against the Merlot database and found that the sample showed a medium level of skin tannin and a low level of seed tannin. When we also benchmarked that against the full variety database, the skin tannin came in with a high reading, seed tannin still a low reading. So for the Merlot, the skin tannin, medium range for Merlot, but in the high range for all red varieties. So with that in mind, the particular year of this study, we were trialing out flash détente as a winemaking tool to see if we could further help extract skin tannin from these Merlot grapes. And so we had different treatments for the extraction of the tannin. The control was just the machine harvested fruit that was just fermented under regular fermentation conditions. Then we had a treatment where 100% of the fruit went through the flash détente system, which is a method, a high burst of high temperature for a short period of time that helps break down the skin cells in the seed and extract things from those skin cells. We also did a mixture of fruit that went through 50% machine harvesting and 50% through that flash détente. And we went through a treatment that had 70% machine harvest and 30% flash détente. What you can see right after fermentation, the machine harvest had a fairly low level of tannin extracted from the skin and seeds in the ferment. The flash détente was more than, well, about 3 times that level of tannin. And with 50% or 70% 50% or 30% flash détente, you can see that the values are a little bit lower than what we saw with 100%. What's important in tannin stability is what's happening months down the road. So 6 months after this initial measurement, we found in the machine harvested fruit, we had lost a great deal of the tannin, whereas in the flash détente treated fruit, the wine still retained a significant portion of the tannin. And similarly, the combined treatments certainly had higher than the control. So Tannin Alert is now available through Covey Analytical Services. We can test skin and seed tannin at harvest for red grape varieties. We have a 3-day turnaround time from sample drop-off to getting your samples. The results will provide not only the skin and seed tannin benchmarked against the variety and the global tannin base, but also tell you if you're in the low, medium, or high range and access the Tannin Alert website. The cost is $100 per sample, and Dr. Jennifer Kelly, my colleague, will be doing a workshop this August for any winemakers that are interested in learning how to use this system. I do have a couple of slides just to thank everybody involved, because this was a multi-year project and lots of partners. So Ontario Grape and Wine Research, Inc. and the Canadian Grape Wine Certification Network. Of course, we couldn't do this work without them. But also early on, when we were brainstorming about how to set up this database and what would be critical information for winemakers, Alan Schmidt, Dave Shepard, Emily Bury, and Craig McDonald were pivotal in that. And then in our last year, doing the flash détente nuance provided the equipment for that. Over the years, we had grape donations for many, many wineries and growers that I've listed here. And then the research collaborators, Dr. Belinda Kemp, worked with me on this project. We had several master's and Bachelor of Science students, lots of technical support. And now Dr. Jennifer Kelly, our enologist in Covey, is going to help with the tech transfer of this program back to industry. And of course, all of our funding partners listed here. And thank you for your time today.
All right, thank you, Debbie. That was a very interesting presentation. I appreciate how you've already done a presentation on, the intricacies of the results back in 2022, which, by the way, is also already posted on CGCN's website under our webinars, so you'll have to scroll for that. But today is just focusing on the services that Covey now has available. I found it interesting that when you were comparing Gamay against other Gamays, it was in that desirable range. But then when you compared it against other red varieties, you see that it actually flops the other way around. Yeah, so this would be really key information for winemakers to bring into their production processes. All right, so I'm going to present our second speaker of the day. We've got Dr. Gavin Kernaghan. He had obtained his Bachelor of Science and Master's of Science from the University of British Columbia before completing his PhD in the Department of Biological Sciences at the University of Alberta. So from there, he held several postdoctoral fellow positions across the country at the University of Alberta, the Université de Québec à Montréal, and the Southern Crop Protection and Food Research Centre at Agriculture and Agri-Food Canada in London. So since 2006, he's served as a professor in the biology department at Mount Saint Vincent University in Halifax, Nova Scotia. His research focuses on the ecology of fungi, particularly the biotic and abiotic factors that govern the structure of fungal communities, with a focus on symbiotic relationships. So I'll let you, Dr. Gavin Kernaghan, take it away with introducing part 1 of your project that was improving wine through fermentations and indigenous yeasts.
Sure, thank you very much for the introduction. Whoops, moving a bit fast. Okay, so I'm going to talk about part of activity 13, the first part of activity 13, improving wine quality through mixed and sequential fermentation with indigenous yeasts. So this is part 1, assessing vineyard yeast communities. So making wine, as we know, usually involves inoculation with the commercial yeast Saccharomyces cerevisiae, which gives us quite predictable fermentations and a predictable product. But there are lots of naturally occurring or indigenous yeasts living on the grape skins that can produce a range of compounds that also influence wine quality. These yeasts can produce alcohols like ethanol and glycerol. They can produce sulfur compounds. They can produce phenolics and hundreds of volatile metabolites, all which can have an impact on the flavor of the wine. So the objectives of this project were to explore native vineyard yeast to find new species to improve wine quality. There are two parts. Part one is assessment of vineyard yeast communities. That's what I'm talking about now. Basically, we need to know what's there, what yeasts are there in the vineyards. And part two, which we're going to hear about from Lihua Fan in a few minutes, is using native yeasts for winemaking. Essentially, how well do these indigenous yeasts work for making wine? Nova Scotia supports a burgeoning cool climate wine industry. We now have 16 commercial wineries at last count in the Annapolis Valley region. There's a map of the Annapolis of Nova Scotia, and there's a blow-up of the Annapolis Valley region. We worked with 8 of these vineyards. 5 of them use conventional vineyard management practices. Those are the dots in red. And the other 3 in yellow used organic vineyard management practices. So this is really the first comprehensive assessment of vineyard yeast communities in Atlantic Canada. So for our sample processing, we took Lacadie Blanc grapes, we collected them at harvest, we pressed them in a sterile fashion, and we let them ferment spontaneously. So we weren't adding any yeasts, we were simply going with the yeasts that were on the skin. And the yeast communities of the initial must, so before fermentation, and also the resulting wines after fermentation, were assessed by next generation DNA sequencing. So we looked at the yeast DNA before fermentation and after fermentation. The relative abundance of yeasts in the vineyard communities were assessed using 2 next generation sequencing platforms, the Illumina MySeq and the PacBio Sequel 2 platforms. They're a bit different. The Illumina MySeq gives hundreds of thousands of short DNA sequences per sample. So it's really good at detecting rare species. It has really good sequencing depth. Whereas the PacBio Sequel 2 gives thousands of sequences, so not hundreds of thousands, but thousands of sequences, but each sequence is much longer, so this gives a better species identification in each case. So we used both of these platforms and we analyzed both data sets to give a really complete picture of the native vineyard yeast communities. So here's our sampling strategy. Between 2018 and 2020, we were interested in variation over time, how the yeast community in a given vineyard changed from year to year. And so we took single samples of grapes from each vineyard each year, and then we split those samples into 4 replicate fermentations, and we did next generation sequencing before and after each fermentation. In 2021, we changed it a bit. We're now interested in within vineyard variation. So how much does the yeast community vary within the vineyard and among vineyards? And so for this kind of sampling, we took 3 random samples from each vineyard. We didn't do any fermentations in 2021, and we sequenced only the musts. We didn't have any post-fermentation sequences for 2021. So here's some results from an example of one vineyard. It happens to be an organic vineyard. In this case, the years and the replicates are pooled. So 3 years pooled together and 4 replicates pooled together. And only species greater than 0.1% of the total community are shown. So none of the truly rare things around here. It's a bit of a complicated diagram, but essentially we've got musts on the top in the black box and wine below it in the gray box. And then you just follow the colored lines to see what the dominant groups are. Some take-home messages from this. Must has a lot more yeast species than wine. There were 159 yeast species found in the musts and only 72 found in the wine. So we lost a lot of diversity while we went through the fermentation. Second point is that must has almost all the non-yeast fungi. So we see this big block of red here. These are all the fungi which are not yeast. These are the filamentous fungi. So these are things that are probably not contributing much to the fermentation process. Third point is that must is dominated by non-fermentative yeasts. So if we look up by must here, we've got Oreobasidium in blue, we've got Phylobasidium in purple, and connected way down here at the bottom, we've got Vishnia Kazaima in green. So these are non-fermentative yeast. They're not making ethanol. This is what's dominating the must. And there are very few fermentative yeasts in must. Very few yeasts that would actually make ethanol are existing in the must. But there are enough of them because the fermentation generally proceeds. When we get to the wine, they take over, and the wine is then dominated by fermentative yeast, Saccharomyces cerevisiae here in teal, and Saccharomyces euverum here in blue. Taking a look at this data in a little bit more detail, I've broken it down into the 4 replicate fermentations. So here in this panel, we have the pre-fermentation data from 2018 on top and 2019 on the bottom. And we see that in all 4 replicate fermentations in both years, it's dominated by the non-fermentative yeast, Oreobasidium pullens here in blue. We've also got, in 2019, we had a good amount of Phylobasidium, also a non-fermentative yeast. And in both years, we had Vishneokozyma, which was also non-fermentative. A little bit more on these non-fermentative vineyard yeasts. They've been pretty much ignored recently, but they are far more abundant on grapes than fermentative yeasts. As I mentioned, there are very few fermentative yeasts in the vineyard yeast community. They may not be involved in alcohol production, but they may still impact wine quality. And that's because they're enzymatically active. They produce volatiles with aromas and flavors like cocoa, almond, whiskey, malt, banana, nutty, pineapple, rose, and honey. So even though they may not be producing ethanol in every case, they are having an impact on the wine flavor. Okay, so continuing with this same figure, we had the pre-fermentation data on the left, and now we've got the post-fermentation data on the right. And we can see in 2018, all 4 of the replicate fermentations were completely dominated by Saccharomyces euverum here in pale blue. And that Saccharomyces euverum is the wild cousin of Saccharomyces cerevisiae, the commercial yeast. It's the wild version of the commercial yeast. In 2019, it was quite interesting. We still had each of the 4 fermentation replicates were dominated by fermentative yeast, by Saccharomyces, but 2 of them in pale blue were dominated by Saccharomyces euverum, and the other 2 in teal colored were dominated by Saccharomyces cerevisiae. So we can see a huge shift here from pre-fermentation with all the non-fermentative yeasts to post-fermentation with those 2 main species of Saccharomyces. So a little bit more on Saccharomyces euverum. This is a non-conventional fermentative yeast. It's common in cool climate vineyards, such as we would have in Canada. Wines made from different strains of Saccharomyces euverum are quite variable in flavor, so there's a lot of diversity there to be tapped into. And one strain is so far being commercialized. So fermentation with Saccharomyces euverum leaves some residual sugar. And so we think it's a good candidate for co-fermentation with Saccharomyces cerevisiae. The idea being that you can start by inoculating with Saccharomyces euverum and make use of the interesting flavors that it gives, and then finish off the fermentation and get rid of the rest of that sugar with co-fermentation by Saccharomyces cerevisiae. So we were also interested in comparing the yeast communities between vineyards with conventional vineyard management practices and those with organic management practices. And this is an ordination diagram. And in a diagram such as this, the points which are closer together have more similar communities. And in this case, they'd be more similar yeast communities. And the points which are further apart have more dissimilarity in their communities. And we can see that the points in white, which are the conventional vineyards, are basically clustering over to the right. And the points in gray, which are the organic vineyard communities, are basically clustering over to the left. So we're getting a separation between organic and conventional vineyards based on the yeast, but it's not the fermentative yeast, it's not the Saccharomyces that you might expect that's making this difference. It's the non-fermentative, or what we call Basidiomycete yeast. And these smaller dots here are telling us what the species are that are actually driving this difference. So for instance, we've got Symmetrospora, Phylobasidium, etc. These are non-fermentative Basidiomycete yeasts that are making the difference between organic and conventional vineyard yeast communities. So just in summary, this is the first comprehensive assessment of vineyard yeast in Atlantic Canada. We found a high diversity of non-fermentative yeast in the must, but this was almost completely replaced by fermentative Saccharomyces species in the spontaneous fermentations. Saccharomyces ovarum can dominate cool climate fermentations, such as in Canada, and they may be a good candidate for co-fermentation with Saccharomyces cerevisiae, which you're going to hear about in a minute. And organic and conventional vineyards differ in non-fermentative yeast species. Okay, with that, I'd just like to thank the Canadian Grapevine Certification Network, the Grape Growers Association of Nova Scotia, Agriculture and Agri-Food Canada, St. Mary's University, and Mount Saint Vincent University. Thank you.
Thank you very much, Gavin, for the presentation. I found it interesting that your non-fermentative yeasts are also still impacting, the flavor profiles of the wine, bringing cocoa, almond, honey, rose, other flavor compounds that you were mentioning. So that's very interesting to learn. We are going to move into our third presentation of the day, which is part 2 of the project that Gavin had started us off with. So now we're going to have Dr. Lihua Fan, who is a food microbiologist working at the Agriculture and Agri-Food Canada Kentville Research and Development Centre in Nova Scotia. She joined AAFC in 1998 and has successfully led or participated in many national and international collaborative research projects. Her research has been focused on food safety and quality, including developing novel natural antimicrobials from plant and bacterial origins for food protection, controlling heat-resistant molds to extend shelf life of processed food products, developing innovative technologies to control post-harvest decay of horticultural produce, ensuring microbial safety or quality of whole and fresh cut fruit and vegetables, and applying fermentation technology to improve flavor and nutrition of value-added products. So Dr. Fan is a professional member of the International Association for Food Protection and the Institute of Food Technologists. She is a certified food scientist and an adjunct professor at Dalhousie University and St. Mary's University in Halifax, Nova Scotia. So with that, please, Dr. Fan, present part 2.
Thank you very much for your introduction. Right. Can you see that?
Yes, we can see your screen for the introduction.
Thank you very much for your introduction. First of all, thanks for organizing this webinar, give us this opportunity. Today, I would like to present the activity 13, part 2, using native yeast for winemaking, following the Gavin's presentation of part 1. Just a very brief introduction. This research is funded by the Canadian Agriculture Partnership Agri-Science Cluster Program. The overall goal for this program is fostering sustainable growth of the Canadian grape and wine. The participants in our activity 13 include AAFC researchers at the Kentville Research and Development Center and researchers from University of Mount Saint Vincent University, Saint FX University, and Acadia University. Our research theme of activity 13 is a focus on native yeast and wine quality. You may ask why native yeast? Because native yeast are very important for the fermentation. They are active at early fermentation stage. They produce alcohol, they influence alcohol content, pH, viscosity, color, phenolics, which are a group of chemical compounds playing a very important role in wine quality. They also produce flavor compounds contributing to the flavor and aroma of the resulting wines. However, currently there has been limited information on native yeast and its community in Nova Scotia vineyard. Given the lack of information regarding the native yeast and the fermentative properties of native cool climate yeast in Nova Scotia, the objective of our research was to isolate, identify, and characterize native yeast strains to control the spoilage yeast and conduct mixed and sequential fermentation using selected yeast strains together with traditional Saccharomyces cerevisiae, and to determine wine quality attribute through chemical analysis and sensory evaluations. By far, more than 300 yeast strains were isolated, identified, and stored in our lab for our long-term research use. Yeast were characterized for their fermentative properties, including alcohol and sulfur dioxide tolerance test, hydrogen sulfide production, enzyme activities. The database of characterization result was created for native yeast and collected in our lab. This slide shows that yeast tolerance tests were done using bioscreen instrument and microplates that generated yeast growth curve for us to determine yeast alcohol tolerance level based on their log stage, lag stage, slope, and the highest optical density values. We selected promising yeast strains, including Saccharomyces euverum and non-Saccharomyces yeast. This slide shows yeast tolerance to alcohol ranging from 0 up to 20%. We measured OD value of yeast growth curve and then calculated that as a percent, where OD at 0% alcohol equivalent to 100% yeast growth. Overall, the 3 Saccharomyces euverum strains had higher alcohol tolerance than that 3 non-Saccharomyces yeast. For example, in this brown color column, more than 94% Saccharomyces euverum can survive alcohol level at 7%, while for non-Saccharomyces yeast, only 9%, 1%, or 15% can survive at this alcohol level. Our selected native yeast produce non or low hydrogen sulfide. Enzyme activities were found in non-Saccharomyces yeast, as you can see from this column in brown color. The Saccharomyces euverum showed high tolerance to sulfur dioxide, growing well in medium with free sulfur dioxide at 60 mg per liter, where non-Saccharomyces yeast survived sulfur dioxide level ranging from 30 to 40 mg per liter. For our fermentation trials, we use selected native yeast. We use commercial available grape juice concentrate as a must. In this presentation, I will show 2 our experimental trials. In trial 1, 3 Saccharomyces euverum isolate were inoculated on day 0, sequentially inoculated with traditional Saccharomyces cerevisiae on day 7 or day 14. In our trial 2, 3 non-Saccharomyces yeast were inoculated on day 0, sequentially inoculated with Saccharomyces cerevisiae on day 4 or day 7. Traditional Saccharomyces cerevisiae EC1118 was served as a control, and fermentations were conducted at 22 degrees for 24 days. The wine samples were collected for quality analysis and sensory evaluations. I just gave a couple of examples about our quality and sensory evaluations. This table is an example of quality parameters of wine fermented with Saccharomyces euverum from trial 1. We determined the alcohol, phenolics, glycerol, different sugars, pH, and acidities in wines. We found that total phenolics contents and the glycerol of the wine were not significantly different among all samples. However, alcohol levels were lower, ranging from 8.9 to 10.2% from native yeast fermented wines, than that in controls of 11.3%. We also found that fructose levels were higher and the total acidities was lower than the controls. These slides show quality parameters of wine fermented with non-Saccharomyces yeast from our trial 2. We can see that the alcohol level were lower, ranging from 6.9 to 9.1%, than those made with controls of 10.8%. Again, total phenolic contents and the glycerols were not significantly different compared with that in controls. This slide shows the correspondence analysis of a sensory evaluation on wine samples from sequential fermentations. This one is from trial 1. The codes in colors group represent the wine samples fermented from native yeast. We use the Saccharomyces euverum yeast, which correlated with fruity and floral profiles, while the C group represented the controls, which correlated with pungent or earthy flavor profiles. This slide is a sensory evaluation result from our trial 2. Similarly, the result was same as the trial 1. 15 experienced panelists found that sequential fermentations using selected non-Saccharomyces yeast were associated with fruit attributes, with more appealing characteristics of citrus, apple, sweet, pear, floral, and tropical fruit. We also found that the timing of the sequential fermentation inoculation with traditional Saccharomyces cerevisiae EC 1118, for example, day 4 versus day 7, or day 7 versus day 14, and the inoculation ratios did not influence the flavor profile of the wines. Our research result demonstrated that native yeast selected from the cold climate wine regions can be used in concert with traditional wine yeast to produce novel, lower alcohol wines with a low risk of fermentation failure. Naturally occurring vineyard yeast can help creating wines with distinctive flavor profile that reflect the local winemaking regions. Our wine fermentation research has been published in the Journal of Plants, People, Planets. Based on our research on yeast community, wine fermentation, and the literature research, we have also completed a review paper discussing native yeast of wine grapes and their potential application to winemaking. This review paper is currently under review by the Journal of Comprehensive Reviews in Food Science and Food Safety. Over the past few years, for our technology transfer effort, we have collectively given presentations at numerous scientific conferences and industry meetings. I just name a few of that include Canadian Institute of Food Science and Technology, the Grape Grower Association of Nova Scotia, Canadian Fungal Research Network, the International Cool Climate Wine Symposium, Last year, we also gave a presentation at the Federal Food Safety and Nutrition Forum and the Kentville Research and Development Center open house. I would like to take this opportunity to acknowledge Canadian Agriculture Partnership Agri-Science Program, Canadian Grape Wine Certification Network, Grape Grower Association of Nova Scotia, researchers at the Kentville Research and Development Center, and our university collaborators. With that, thank you very much for your attention.
Thank you, Dr. Fan, for the presentation. I also want to take this opportunity to remind everybody that if you do have questions for any of the presentations, we have one more, but if you have any questions, please feel free to put them in the chat section at the bottom of your Zoom screen, or save them and raise your hand and we'll give you access to your mic so you can ask them in live time. So I don't need to present our next speaker because they have already presented, but Dr. Debbie Inglis is going to present on behalf of Dr. Belinda Kemp, who can't be with us today to present on her research. But please take it away to present improving still and sparkling wine qualities, Debbie. Thank you.
Thank you. Okay. Thanks, Darian, and you can see this slide okay?
Yep, all good.
Okay, great. So yes, I'm presenting my portion of the work that Belinda and I did on the sour rot honey compound sparkling project in the CAP program. And the outputs from this program have continued on into SCAP. So I'm kind of bridging both the CAP outcomes and the SCAP outcomes. So today I'm going to talk about preventing high volatile acidity in wines using natural Canadian Indigenous yeast isolates. So great wines, I think we can all agree, begin in the vineyard with quality fruit. However, winemakers still require additional enological tools. And they need these tools to further enhance the aroma profile of wines, to differentiate wines in the marketplace. And I think that's critical here. I think that's why so many in Canada are looking to look at their Indigenous yeast populations and start to use them to help to differentiate wines in the marketplace. And then we also see that winemakers need tools to overcome challenges from Mother Nature. So these tools can include non-traditional yeast strains that alter and improve the aroma profile of wine through the volatile organic compounds that they release, as well as some other core compounds from the fermentation. Yeast may also help mitigate the impact of fruit rots, such as botrytis and sour rot infected fruit. So there is a growing interest worldwide, including in Canada, to further differentiate wines by expressing a region's microbial terroir. And I think we just saw a great presentation combination from Gavin and Lihua to really showcase how that can be implemented. The use of natural yeasts on fruit to ferment wine, is an area of research we're all embarking on, and it can add complexity to wine due to the contributions of many different yeast species. However, there are some risks with just going with a natural spontaneous fermentation. There can be inconsistencies that result in fermentation to fermentation. There may be off flavors and oxidation faults if you haven't characterized the individual yeasts that are in your fermentation, isolated them, and then back inoculated with the ones that are the good guys versus the bad guys. So most commercial yeast on the market are of the species Saccharomyces cerevisiae, although there are a handful of non-Saccharomyces yeasts that are entering the market. And as Gavin mentioned, there's one Saccharomyces euvarum that is now also on the market. Saccharomyces cerevisiae, why is it so popular? Because it's alcohol tolerant, it can complete the fermentations, and the strains that are on the market have been selected for their enological performance and flavor profile of the resultant wines. And that information is shared with winemakers so that they can choose the yeast that's best suited for the variety that they're fermenting. Most Saccharomyces cerevisiae yeasts, however, were selected from European countries and may not be the best performing yeast for our cool climate regions. Hence the research that all of us today have been focusing in on. So commercial yeast, the wines may not exhibit characteristics of the local microflora if you're dominating the fermentation right off the start with a commercial yeast. However, there is more control over the ferments and less risk of step fermentations and off flavors due to unwanted yeast strains. Some winemakers, however, with spontaneous fermentation, like the complexity of working with indigenous natural yeast, but not all indigenous yeast can complete a fermentation, and many can produce off flavors. And it can be more difficult to control a natural spontaneous fermentation because you're not sure what yeast is going to be present. But what if we could isolate our own yeast and inoculate with them so that they dominated or contributed to the fermentation? And you saw some applications of this in the previous presentation with fermentations with a single yeast or co-fermentation results. So I want to start by just sharing some past research that has been carried out in my lab using a locally isolated Saccharomyces euvarum yeast that we called CN1. And we started really investigating this yeast for commercial application with appassimento wines. So that's wines made from red grape varieties that have been further concentrated off vine. This particular yeast, CN1, that we've been working with for a number of years now, was isolated from Riesling ice wine grapes. And I really wanted to see if it could assist with lowering volatile acidity that can result in wines made from fruit that is more concentrated at higher Brix level to begin with. So again, I presented some of this work in the past, so I'm just going to focus in on some of the main components here and just trying to see if you can you see my pointer here?
Yeah, I can see it.
You can see it. Okay, great. Whoops, too far. In this set of fermentations, we had grape juice that varied from 21.5 Brix to 24.5, 26, and all the way up to 27.5 Brix. And this was a juice of Cabernet Franc that had been further concentrated through a drying process to get to those high Brix levels. And what I want you to take from this series of graphs is that throughout the fermentation, when we monitored the amount of acetic acid being generated through the fermentation versus time, we see time and time again for the ferments supported by CN1, the red dots, always the acetic acid was lower in the final wine than what we saw for EC1118. But also, as that Brix concentration increased, we saw this phenomenon where the yeast would start to produce acetic acid, but then the acetic acid level would drop, meaning that they were consuming acetic acid at a faster rate than what they were producing it. And we saw this drop, and then it kind of leveled off as the fermentation continued. And this always resulted in a much lower level of acetic acid in these wines compared to the control Saccharomyces cerevisiae EC1118. What we also found with this yeast was a very different aroma profile in the wines that resulted in the CN1 supported fermentations versus the EC1118. I'm not going to go through all of these compounds, but I think you can see that the pattern of these bars in these graphs is very different for CN1 versus what we see for EC1118. And the CN1 wines had a lot of fruity and rose compounds that resulted in the wines, but less banana, strawberry, and green apple compounds. If I could just do a quick summary there. When we did the sensory profile, a detailed sensory descriptive analysis, we found that these wines with CN1 increased the black fruit aroma and flavor and reduced the sourness and astringency in the wines compared to EC1118. And we also carried it a step further and looked at the consumer preference of these wines and found that the wines fermented by CN1 were liked just as well as the wines fermented with EC1118. There's actually no significant difference in these liking scores. So the consumers were liking the wines fermented with this yeast, and this yeast was reducing the volatile acidity and some of the challenges of oxidation components that can result in high sugar ferments. We decided to work with Lallemande to make an active dry form of this yeast and test it on a commercial scale. And what we found on the commercial scale is just as we had found in our trials in our research winery, the wines fermented with CN1 always had lower levels of acetic acid present in them versus the wines fermented with EC1118. I'll also point out for each of these wines, although I haven't shown you the fermentation data, I'll show that to you for some subsequent ferments. The ferments always went to dryness. So this Saccharomyces euvarum isolate ferments to dryness, doesn't leave residual sugar. So one of the drawbacks with making an active dry preparation of this yeast is that it has a very high death rate. It doesn't undergo the active dry process that well. And so we've been investigating another way to prepare the yeast in wet culture, which would really be conducive for any yeast that local wineries are wanting to isolate and further put into a form that they can grow up and scale up for their commercial operation. So this is going to lead into more of the recent work that we've been doing under SCAP, which is a continuation of this work to look at the ability of CN1 and other Saccharomyces euvarum strains to overcome issues of botrytis or sour rot infected fruit, where that fruit, when it comes into the winery, can have high levels of acetic acid, ethyl acetate, ethanol, acetaldehyde, glycerol, galacturonic acid, and gluconic acid. But what the wineries typically measure in the fruit coming in with botrytis and sour rot is the volatile acidity. And in Ontario, when that VA gets above 0.2 to 0.24 gram per liter, the wineries typically reject that fruit. But here we had a yeast that we were investigating in the Appassimento project that showed that it could consume acetic acid and result in lower acetic acid in wine. So we wanted to trial this yeast, the Saccharomyces euvarum CN1 yeast, to see if it could help with white wine fermentation that had been impacted with sour rot. We had done a lot of work previously in red wine ferments, but we had never trialed this yeast in white wine ferments. So we set up a fermentation scenario where we had fruit that was 0% infected with sour rot and botrytis. We did mixtures, so it was 20% infected with sour rot botrytis fruit and 40% infected with sour rot fruit. And we carried out fermentations with both EC1118, our controlled Saccharomyces cerevisiae yeast, and our Saccharomyces euvarum yeast, CN1. And we carried out all these fermentations in triplicate. Just focusing first on the sugar consumption. What I've got plotted here is the soluble solids, or the Brix level on the side, and the days of the fermentation below. And we found in each case that our fermentations did go to dryness, and in actuality, the CN1 always was a little bit ahead of the EC-1118 midway through the fermentation. But then by the end of the fermentation, it often required 1 or 2 extra days to complete that fermentation and go to dryness. And for that fruit that was at the control, the 20% or 40% infection with sour rot fruit, that resulted in 0 grams per liter acetic acid in the starting fruit, 0.2 grams per liter in the starting fruit, or 0.4 grams per liter, which would have been well past the acceptable range in the starting fruit. Throughout the fermentations, we also wanted to make sure, because there were a lot of microbes that were present in, especially in the rot treatments, we wanted to ensure that our CN1 yeast, along with the Saccharomyces cerevisiae EC-1118, were implanting in these fermentations and still dominating the fermentations. So although, as Gavin pointed out, there are more advanced techniques to identify these yeast species, we wanted just something quick and cheap that we could follow throughout the fermentations. And so we did a plating assay on some media that would change color depending on what yeast was growing on it. If it was Saccharomyces euvarum, we got green colonies when the yeast first started to grow. And if it was EC-1118 or cerevisiae, we got cream-colored colonies. So we could follow the progression of the yeast through the ferments. So when we got the initial Riesling must, we found that all of the yeast in the starting must, they were not Saccharomyces cerevisiae or euvarum that we could detect with our plating. They were other yeast species. Early in the fermentation, EC-1118 dominated Saccharomyces cerevisiae dominated the ferments that were inoculated with EC-1118, and you can see that by all these blue bars. Whereas the CN1 inoculated yeast were all dominated by Saccharomyces euvarum. Now, at this stage, we're assuming that the cerevisiae is EC-1118 and the euvarum is CN1. We have gone back and verified that using more molecular approaches. Midway through the fermentation, we still see cerevisiae dominating the cerevisiae and CN1 dominating, or euvarum dominating the CN1 inoculations, and this held through right to the end of fermentation. And we saw, not sure if you can pick this out, but in the 0.2 gram per liter acetic acid ferments with EC-1118, there's a little bit of euvarum starting to show up in that fermentation at the end. The CN1 ferments stayed dominated by euvarum. Looking at the acetic acid profile in the must versus the wine, this is where we saw some very drastic differences between the wines fermented with CN1 in orange and the wines fermented with EC-1118 in blue. So again, each of the musts, they had a level of sour rot and botrytis infected fruit. So they started out at very low levels of acetic acid at 0.2 or at 0.4. For the initial must, but look at the ferments inoculated with CN1. The final wines had a drastic drop in acetic acid, coming in at less than 0.08 gram per liter acetic acid for the worst case scenario of the wines with the 40% rot treatment. Versus what we see with EC-1118, EC-1118 produces more acetic acid than what was present in the initial must for the 0 and the 20% treatment. And then we did see a little bit of a drop in the high treatment with the 40% rot present. As Gavin and Lihua pointed out earlier, We also found that the ethanol levels in the wines fermented with CN1 were always a little bit less, usually about 0.5% less in these Riesling ferments compared to the EC111A. But what we found in exchange for that lower level of ethanol, the yeasts were producing higher levels of glycerol in each of these cases with the CN1. So again, all of these ferments went to dryness. So the sugar is getting diverted instead of to ethanol, it appears it's going to glycerol. We saw no difference in the acetaldehyde between the yeast strains. We carried this further on looking at the volatile aroma compounds, and we found that the wines fermented with CN1 produced much higher levels of higher alcohols, specifically 2-phenylethanol, which smells like rose. It's a very floral attribute in wines. We found that CN1 produced lower levels of acetate esters, and there was a different profile of ethyl esters that resulted depending on whether the wines were fermented with EC111A, the first three columns here, or if they were fermented with CN1. So we decided to continue in the SCAP with looking at additional Saccharomyces euvarm strains. And we had worked with a local winery in the previous years that we found, and of those 12 euvarm isolates, there were 6 different strains of Saccharomyces euvarum. So this project, we wanted to profile what was happening in Chardonnay juice fermentation if we inoculated with those 6 different Saccharomyces euvarum yeasts, along with CN1, our euvarum that we've been working with for a long time, and in comparison to EC111A. What we found here, using the Chardonnay juice, it came in at about just over 21 brix, fairly typical parameters here for this juice, TA of 6.2. The total nitrogen was a little bit low, only at 126 milligrams of nitrogen per liter. So we decided in the ferments that we would supplement with nitrogen at various time points. And in the fermentation graphs here, you'll see the points where we added nitrogen in the form of Fermate O, which is an organic form of nitrogen, or diammonium phosphate at these different time points throughout the fermentation. And this was just to ensure that the yeast had more than sufficient nitrogen to carry through their growth and metabolism right through to the end of ferment. Again, we found that all of the yeasts went to dryness. We do have residual sugar measurements on all of these. So all of the sugar was consumed through the ferment. EC111A, in this case of the Chardonnay juice, completed the fermentation earlier than any of the euvarm strains. But really, the euvarm ranged from taking 1 more day up to 3 more days to complete the fermentation, but they did all complete. We followed the implantation again through the fermentations, evaluating the yeast profiles early in the fermentation, midway through the fermentation, and at the end of the fermentation to make sure that the inoculated yeasts were still present at the end of fermentation. And all test strains did implant into the fermentation and successfully completed the fermentation. We do see that there is some cerevisiae, the blue bars, showing up in the fermentations later on, but they're still dominated by the euvarm yeast that were inoculated. Just looking at the ethanol profile again, we found some of the euvarm strains produced significantly less ethanol than our control EC111A. And one of these strains, we call it CS6, it produced the ethanol production was over 1% less than what we were finding with EC1118. And we're very interested in these lower ethanol producing strains because there is a movement in the industry to go to lower alcohol wines. And we're continuing to evaluate the purpose of these yeasts for lower alcohol wine production. Similarly to what we saw with CN1, even though the yeasts are producing less ethanol, they're producing much higher levels of glycerol than the control yeast, EC1118. And so where EC1118 was producing, let's say, about 6.8 grams per liter of glycerol, we see some of these strains producing about 11.8 grams per liter glycerol. So quite a large jump there. We also see all of these strains producing lower levels of acetic acid, which initially was why we were evaluating these strains to look for control points for fruit coming in with sour rot. Bench trials of the wine aromas and flavors, just from our bench trials. So this isn't formal sensory evaluation yet. We just wanted to document, what these wine aromas and flavors were. So we had a panel of 5 people just giving their impressions of these wines. And I think, as you can see, for all of the descriptors, these are, for the most part, quite pleasant, except for maybe the sweat attribute on one of these yeast strains. So we're quite interested to move forward with larger scale ferments using these yeasts on a commercial production, where we will carry out a more formal sensory evaluation on these wines. So we plan to trial some of the Saccharomyces euvarum strains from this project at the commercial winery this fall using Chardonnay. We are now working with a local yeast company here in Ontario. They're called Escarpment Labs to grow up the yeast for commercial trials in a wet paste format. So since it's not easy to actively dry these yeasts, we need another way to grow them up in sufficient volume and concentration that they can be easily used at wineries. This yeast company, Escarpment Labs, works closely with the brewing industry, and many brewers do not use active dry forms of yeast. They use these paste forms of yeast. So we're just converting that technology for application in wine production. All the VOCs and volatile fatty acid analysis has been completed on all of our research wines. I just didn't have time today to delve into all that, but we have presented that recently at a conference this summer at the American Society of Enology and Viticulture Eastern Section. And the differences between strains in the VOCs and VFA analysis will help us determine which strains we want to move forward with for the commercial scale up. The winery doesn't have capacity to do all of the strains that we've isolated from their winery, so we're going to pick and choose which ones we want to scale up. So in summary, Saccharomyces euvarum strains can be beneficial to enhance the aromatic profile of wines produced from sound fruit, providing a unique identity to the wine for market differentiation. Saccharomyces euvarum CN1 continues to show application for use in botrytis sour rot infected fruit by reducing acetic acid in wines while enhancing the volatile aroma profile of those wines. And Saccharomyces euvarum may prove useful in reducing alcohol from 0.5 to 1% volume per volume in wines by diverting sugar to compounds other than ethanol. And our future research will continue to work with the yeast company on the grow-up procedure for the wet cultures or paste cultures for industry. And with the commercial ferments that will start this fall, we'll be carrying out the sensory descriptive analysis on these wines, as well as all of the VOC and VFA analysis, so that I can report back to you maybe in a couple of years on the outcome from that. So with this work, I'd like to thank my research colleague, Dr. Jen Kelly, who's working with me now on all of these trials, and my former collaborators, Dr. Gary Pickering and Belinda Kemp, that worked on the Appassimento and the some of the early work with CN1. Fei Yang and Sufan Zhu are technologists here in Covey that do a lot of the analytical analysis. I've had several master's students, Daniel Philippou and Frederick Rivard, that are currently working on their MSCs. Daniel here at the end picture is just getting ready to defend this summer, and Fred still has another year to go. There is a fourth-year thesis student involved. Lots of industry partners, Seeger Vineyards, Nomad Winery, King and Victoria, and Piloteri, that donated all of the grapes for these projects, Cave Spring Vineyards and Piloteri for the commercial wine production. And then, of course, our funders, Ontario Grape and Wine Research, Inc., Agriculture and Agri-Food Canada, and the Canadian Grapevine Certification Network. So thank you very much for your time today.
Thank you, Debbie. Well, we've got some questions coming in from the audience, so I think now would be a great time to have our Q&A session, and I'll moderate that. So, for all the presenters, if you could, turn on your cameras, have your audio on. The first question is, how are producers of your respective regions responding to these new yeast isolates and or to sequential fermentations? And how is the reception from the industry?
I can certainly start with that because it was the industry that allowed us to further expand our evaluation of some of these strains. The industry here in Ontario is really looking forward to trialing their own yeasts in their own fermentation. And again, part of this is for market differentiation. They want to be able to tell the microbial terroir story of their vineyard operations. The sticking point is figuring out a way to grow up those yeasts in a large enough volume that they can be used in commercial fermentations. And that's why I've brought in Escarpment Labs in Ontario to work with us on that step. How can you develop a yeast preparation in an easily accessible manner that can be developed, a couple of weeks before you need them and are going to have viability, through the commercial ferments. So from my end, I'm asked all the time when CN1 is coming onto the market, and I am just meeting with Escarpment Labs later this week to talk about having them be our commercialization partner for this, since the yeast, it's not feasible to undergo the active dry process. You need 5 times the amount of yeast for an inoculation, and it's just not cost effective.
Mm-hmm. Was there anything else that Gavin or Lihua you want to add to that?
Maybe I can put on some comments here.
Mm-hmm.
And because our research was conducted at lab scale, so we haven't had the chance to test at the commercial scale to use this native yeast that we found that is promising. But I'm very glad to hear that Debbie mentioned in the Ontario, and there's a company to develop that start culture and potentially to use that. For us, we didn't have a chance to participate in the second round of Grape Wine Cluster project, so we didn't have the chance to further develop this start culture. But when we have the meeting with talking about our research, we got some good feedback. They really welcome this idea. Also, there's consumers, they prefer their lower level of alcohol. They do want to have the good flavor, but a lower concentration of alcohol product.
Mm-hmm. And actually, the next question from the audience is actually touching on low alcohol as well. They're wondering if there's interest from the wineries in low alcohol beverages, or is it mostly just research interest for now?
No, there's definitely interest from the wineries. Some of the wineries here in Ontario are actually partnered with wineries from other parts of the country and importing low and no alcohol wines. Some wineries are making lower alcohol wines, but you know, we feel that we can bring a different tool to the table by the yeast itself, you know, that where they divert some of their sugar metabolism away from ethanol and more towards glycerol, you know, from our initial analysis. And, you know, if we can, you know, get wines that are 1.5% lower in that level just from the yeast selection, then we also have lots of research ongoing, looking at different varieties that start at ripeness with lower sugar levels, so that, you know, the potential alcohol is lower right off the start, then using the right yeast strains and, you know, if need be, some technology like reverse osmosis to get it down, you know, even lower. So definitely there's industry interest. I would say that research is a little more preliminary. We've just started on that this past year. So there'll be lots more to come in the future.
Mm-hmm. Well, I have one more question, unless something else comes in from the audience. Are there sprays or are sprays impacting indigenous yeasts, and more specifically, some growers are using peroxide, so would that have effects on the yeasts?
I
Shall I
don't know,
say?
Gavin, do you want to try to
Sure.
take that one? Because
I
you've
can
got
say
more
something on
population.
that. Yeah, I've heard about the peroxide. We don't have any experience with that here in our research, but the differences we were seeing between the organic and the conventionally managed vineyards seems to be the result of sulfur application. And some of these Basidiomyces yeast, these non-fermentative yeasts, have more or less tolerance to sulfur. So that seemed to be what was behind that.
Can I just make one comment, Darian? I should point out to you, for all of the work that we've been doing with the strains that we've selected, we always isolate them from the end of the fermentation. You know, it's not that we couldn't isolate them from earlier on, but our thought process was we wanted to ensure we were using yeast that could complete a fermentation. That may, you know, cut out a whole whack of yeast that could, you know, add other flavor and aroma compounds to the wine. But our initial trials were to isolate from the end of the fermentation. So just to put that into perspective too, we're not looking at yeast. It would probably be really hard to find our yeast at the start of fermentation in a spontaneous one because the populations are so low at that point.
Right. Okay, well, unless there's any other questions, maybe Ethan, I'll just check in with you to see if there's anything else on your end of things.
I have one more comment.
Oh, yeah, sorry, go ahead.
Yeah, sorry. I think the question earlier was, are wineries interested in lower alcohol wines? But what I want to also add to that is that consumers are interested in lower alcohol wines, and there's, you know, a significant interest level from the consumer component on lower alcohol and zero or de-alcoholized wine. And we want to make sure that our industry across Canada doesn't miss out on the boat in those categories. Absolutely. By making really good quality lower alcohol and reduced or zero alcohol wines. Hence, you know, the research that we're doing. So the consumers, you know, have a lot of clout in terms of, you know, what they're going to choose at their various locations for purchase. So we want to make sure that, you know, we're ready as an industry to fill that marketplace. You know, I'm sure everybody is seeing on the grocery stores, you know, the low or zero alcohol wines on the shelves. And we think we can produce a better quality product with local Canadian grapes that get fermented, but, you know, still capture all of the wonderful aroma and flavors of a wine, but, you know, the reduced or no alcohol level in those wines.
Yeah, thank you for adding. And actually, in the midst of you kind of answering that or adding to that, we had another question come in about low alcohol. So what about climate change, the impact on that? Won't hotter weather have an impact on the low
Well,
alcohol?
it potentially could, especially if you're using varieties that further increase the sugar content, those warm seasons, but also evaluating new sustainable varieties coming into the marketplace and certainly coming into Ontario so that we can assess their viticulture performance under our environmental conditions. And these grapes are at ripeness at lower Brix level, 18, 19 Brix. So already you're taking, fruit that maybe in the past we were fermenting at 22, 23 Brix. These varieties just don't get to those Brix. They're fully ripe at a lower Brix level. And we want to see if they could be contenders for these lower alcohol or de-alcoholized wines.
Thank you, Debbie. Gavin or Lihua, did you have anything else to add to the low alcohol or no alcohol questions?
I agree with what Debbie. I agree with what Debbie said.
We'll go with Lihua first.
Just to follow up Debbie's comment, I think for the climate change questions, generally speaking, for our fermentation part, if the weather increase, let's say temperature increase 0.5 or 1 degree, perhaps it may not impact that so much. However, if the extreme temperature happened, for example, the frozen at early spring, that definitely impacts the quality of the wine product, or if the smoking. So that is a lot of other issues. Definitely a lot of research needs to be done in that area too.
Yeah, absolutely. Gavin, do you want to add to that?
No, I was just agreeing with Debbie.
Oh, okay. Sounds good. And then we have one last question, and then we will close up the session for today. How common are co-fermentations in commercial production? I guess this would depend on your area as well.
Yeah, I mean, I think there are certainly yeast products out on the market that are being promoted for co-fermentations. I personally haven't done a survey, within the industry. It would be, one more step, especially if you have to start a fermentation with one yeast and then add a second yeast. And you have to have the manpower and you have to be following your ferments to be able to do that. But at the end of the day, if it developed or produced a wine of superior quality that you could differentiate in the marketplace, it might be worth that effort. So, I think depending on where you're located, the size of your operation, just, the manpower you have to do that sort of tracking would dictate whether you would take those extra steps.
Thanks, Debbie. Gavin or Lihua, can you comment on that from the East Coast perspective? On co-fermentations?
Go ahead, Lihua. I would only say I don't think anybody is doing co-fermentation at an industrial scale in Eastern Canada.
Yeah, I think only some wineries doing this natural fermentation. I guess there's one or two, but not a co-fermentation. I think for the research point of view, that is important to add in co-fermentation or sequential fermentation and to produce good quality and more flavorful wines. But at this point, in our region, we don't have that yet.
Do you find then that natural fermentations are pretty successful in your regions?
Currently, yes, there's some successful stories for the natural fermentation, but we did research. We collected the samples, grape samples from different vineyard, and when we conducted the spontaneous fermentation, we found some site the samples couldn't go through the fermentation because the initial yeast they couldn't initiate the fermentation, but some site the samples can go half down fermentation. So there's a risk for the fermentation failure of that case. So we need to consider a different way to do the sequential fermentation.
There are also award-winning wines from Nova Scotia that are purely spontaneous fermentations. So they can work really well when they do work.
I think it's also important to note that often, may not be the case for everyone, but often when they're doing a spontaneous fermentation, they will take a portion of that juice and start what we would call a starter culture, let it start to build up the microbial population. And then they, by the time that kind of is halfway through a ferment, or the sugar drops so far, you've got a larger population now established in that starter culture that are the fermenting yeast. And then that is used to inoculate the rest of the ferment. And that leads to more successful spontaneous fermentations. They're still spontaneous. It's still just the microflora that was in the juice, but you give the opportunity on a smaller portion. Maybe you pick the grapes a couple of days before and you get that starter culture going, and then you inoculate into the rest. And that gives you a little more control over those fermentations.
And Saccharomyces cerevisiae can also have a lot of inertia. So you may think you're doing a spontaneous fermentation, but you're really fermenting with the yeast that you used in previous years. It's still around in the equipment.
Okay, well, I think that wraps up our session for today. I realize we went a little bit over time, but I appreciate everyone for staying a little bit longer. So I want to thank our guest speakers, Debbie, Lihua, and Gavin. Thank you for sharing the final results from these important research projects, and we look forward to, in some cases, continuing to work with you, or hopefully having to work with you in the future to strengthen and improve Canada's grape and wine industry. And to everybody that's joined us today, I want to thank you for joining us for probably going to be our last webinar of 2025, but you will see us again start up for 2026. We'll bring another mini series or another series to light. So stay tuned on our social media channels on Facebook, X, and LinkedIn, and our website to be informed of upcoming news and events. And if you've enjoyed this webinar or other cluster updates that we've had so far, and you want to check out the research updates recordings, then you can see the AAFC research results under the CAP cluster that was hosted on April 17th. And then there's other cluster research updates that we've provided throughout this 2025 as well. So you can find everything there on our website and our YouTube channel. Again, this webinar has been recorded, so it's going to be posted to our website and our YouTube channel, hopefully by end of this week or early next week. And lastly, when you exit this webinar, you'll be asked to complete a very brief survey where you can share your thoughts on today's sessions. We really appreciate if everyone could take a couple minutes to fill this out so we can improve our sessions for the future. So thank you very much to our speakers. I hope everybody enjoys the rest of their day, and we'll see you for more webinars in 2026.
Thank you.
Bye.
Thank you.
Thank you.